Welcome to Sleepy Science Odyssey. Tonight, mushrooms, the visible fruiting bodies of fungi, organisms neither plant nor animal, from the familiar button mushrooms in grocery stores to the vast underground networks connecting forests, from decomposers breaking down dead wood to symbionts partnering with tree roots, from bioluminescent species glowing in the dark to toxic varieties that can kill with a single bite. We'll explore fungal biology, ecology, evolution, and the remarkable ways these often overlooked organisms shape ecosystems, enable life as we know it, and demonstrate strategies for survival fundamentally different from plants and animals. If you're new here, this channel is for people who need something to think about before they can stop thinking. Real mycological science, no exaggeration, just quiet facts in the dark. Let's begin. Fungi comprise a biological kingdom separate from plants, animals, and bacteria, with an estimated 2.2 to 3.8 million species globally. When you see a mushroom growing from soil or wood, you're observing only a small, temporary part of a much larger organism hidden beneath the surface or within the substrate. The mushroom itself, the umbrella-shaped structure with a cap and stem that most people recognize, is merely the fruiting body, a reproductive structure produced by the fungus to disperse spores, analogous to an apple on a tree rather than the tree itself. The main body of the fungus consists of hyphae, thread-like filaments typically only a few micrometers in diameter, far too thin to see individually with the naked eye, that grow through soil, wood, leaf litter, or other substrates, branching and extending in all directions to explore the environment for nutrients and water. These hyphae collectively form a network called a mycelium, a term derived from the Greek word for mushroom. A mycelium can extend over vast areas. Some of the largest known organisms on Earth are fungal mycelia, with one individual of the honey fungus Armillaria osti in Oregon's Blue Mountains estimated to cover approximately 965 hectares, nearly 10 square kilometers, and weigh hundreds of tons, possibly exceeding 35,000 tons based on estimates of hyphal density and extent. This individual, informally called the humongous fungus, may be 2,400 to 8,650 years old based on growth rate estimates, making it not only one of the largest, but also one of the oldest living organisms. The mycelium grows through forest soil and attacks tree roots, causing root rot disease in conifers, and its extent was determined by analyzing genetic samples from fungal tissue collected across the area, revealing that tissue separated by kilometers was genetically identical, indicating a single individual rather than multiple separate organisms. The hyphal structure provides fungi with remarkable efficiency in exploring substrates and extracting nutrients. Individual hyphae are tubular cells surrounded by cell walls composed primarily of chitin, the same tough polysaccharide that forms insect exoskeletons and crustacean shells, rather than cellulose found in plant cell walls. This chitin provides strength and flexibility, allowing hyphae to penetrate substrates by growing through tiny pores and cracks, exerting pressure at growing tips, and secreting enzymes that soften and dissolve materials blocking their path. As hyphae grow, they branch at regular intervals, creating an expanding network that maximizes contact with substrate, allowing the fungus to explore large volumes and encounter nutrient patches that would be missed by simpler growth patterns. The hyphal network operates somewhat like a circulatory system, though without a heart or central pump. Nutrients and water absorbed anywhere in the network can be transported throughout the mycelium through cytoplasmic streaming, active movement of cytoplasm within hyphae driven by molecular motors moving along cytoskeletal tracts. This allows fungi to translocate resources from nutrient-rich areas to nutrient-poor areas, supporting growth into new territory using resources extracted elsewhere. The efficiency of this transport has been demonstrated experimentally. When parts of a mycelium are supplied with nutrients while other parts are growing through nutrient-poor substrates, the supplied nutrients rapidly appear throughout the network, supporting growth at the nutrient-poor frontier. This resource translocation allows fungi to act as conduits, moving nutrients across distances in ways that have important ecological consequences, particularly for mycorrhizal fungi that connect plants and can transfer nutrients between them. Fungi are heterotrophs, meaning they must obtain organic carbon from other organisms rather than producing it themselves through photosynthesis like plants. However, unlike animals that ingest food and digest it internally in specialized digestive systems, fungi digest their food externally by secreting enzymes into their surroundings, then absorbing the resulting small molecules through their cell walls and membranes. This external digestion allows fungi to consume food sources they cannot ingest: solid wood, tough plant material, soil, organic matter, by dissolving them in place. The enzymes fungi produce are remarkably diverse and powerful, including cellulases that break down cellulose, lignin peroxidases that degrade lignin, proteases that digest proteins, lipases that break down fats, and many others specific to particular substrate compounds. The ability to produce this enzymatic arsenal, particularly enzymes that degrade lignin and other recalcitrant compounds, makes fungi crucial decomposers in ecosystems. Lignin, the complex polymer that stiffens plant cell walls and comprises 15 to 40% of wood by weight, is extremely resistant to breakdown. Most organisms lack the enzymes necessary to degrade it. Fungi, particularly basidiomycetes, including many mushroom-producing species, produce powerful oxidative enzymes, including lignin peroxidases and lacases, that can break lignin's irregular, cross-linked structure. Without fungi performing this decomposition, dead wood would accumulate indefinitely, locking up nutrients and carbon rather than recycling them back into ecosystems. Indeed, fossil evidence suggests that wood accumulated in massive quantities during the Carboniferous period, approximately 300 to 360 million years ago, forming the coal deposits we mine today, possibly because lignin-degrading fungi had not yet evolved or had not become widespread, allowing dead trees to accumulate and become buried rather than decomposing. Fungi are classified as a separate kingdom of life, distinct from plants, animals, bacteria, archaea, and protists, based on multiple fundamental differences in cell structure, biochemistry, development, and evolutionary history. DNA sequence analyses confirm that fungi are more closely related to animals than to plants. The fungal and animal lineages diverged from a common ancestor approximately 1 to 1.5 billion years ago, while the plant lineage diverged from other eukaryotes even earlier, over 1.5 billion years ago. This means humans are more closely related to mushrooms than mushrooms are to lettuce, a surprising fact that seems counterintuitive given superficial similarities between fungi and plants, like growing in soil and being stationary. The shared ancestry with animals is reflected in some cellular and biochemical similarities, Both fungi and animals store energy as glycogen rather than starch. Both produce chitin in some structures, and both lack chloroplasts and photosynthesis. The diversity of fungi is staggering, but largely hidden from casual observation. Scientists have formally described approximately 150,000 fungal species, but this represents only a small fraction of actual diversity. Estimates of total fungal species numbers vary widely. Some molecular studies analyzing environmental DNA suggest 2.2 to 3.8 million species may exist globally, while more conservative estimates based on fungus-to-plant ratios suggest 1.5 million species, and some mycologists argue for even higher numbers, perhaps 5 to 6 million species. Regardless of exact numbers, the consensus is that the vast majority of fungal species remain undescribed, unknown to science, awaiting discovery. Many of these undescribed species are microscopic molds and mildews that never produce large fruiting bodies and are difficult to culture in laboratories, making them challenging to study using traditional mycological methods. Modern molecular techniques using DNA sequencing of environmental samples are revealing hidden fungal diversity, with studies routinely detecting hundreds of distinct fungal sequences in single samples of soil or water, many not matching any known species. Fungi occupy every terrestrial environment and many aquatic environments, from Arctic tundra to tropical rainforests, from desert soils to deep ocean sediments, from inside living plants and animals to the surfaces of rocks exposed to full sun. This ecological breadth reflects metabolic versatility and stress tolerance. Some fungi can grow at temperatures below freezing, while others thrive in hot springs above 60 degrees Celsius. Some tolerate extreme desiccation, surviving for years without water, while others require continuously moist conditions. Some can grow in saturated salt solutions or in the presence of heavy metals toxic to most organisms. This versatility allows fungi to fill ecological roles unavailable to organisms with narrower tolerances, making fungi essential to ecosystem function globally. Mushrooms are the spore-producing fruiting bodies of certain fungi, analogous to flowers on plants. When environmental conditions are favorable, usually when adequate moisture, appropriate temperature, and sufficient nutrients coincide, the vegetative mycelium of certain fungi produces fruiting bodies. The structures we recognize as mushrooms, toadstools, brackets, puffballs, morels, and countless other forms. These fruiting bodies serve a single primary function: producing and dispersing spores, the microscopic reproductive units that will give rise to new fungal individuals if they land in favorable locations. The timing, frequency, and abundance of fruiting body production varies enormously among species and with environmental conditions. Some species fruit annually or even multiple times per year if conditions permit, while others fruit sporadically, going years or decades between fruiting events. Some produce small, short-lived fruiting bodies that last only days or even hours, while others produce massive, woody structures that persist for years or even decades, continuing to produce spores season after season. The familiar mushroom form, a cap, technically called a pileus, atop a stem, a stipe, represents just one of many fruiting body architectures fungi have evolved. This agaricoid form is common in the order Agaricales, which includes many familiar mushrooms like the button mushrooms sold commercially, the deadly Amanita species, the luminescent Armillaria honey fungi, and thousands of others. The cap typically bears gills on its underside, thin, vertical plates radiating from the stem to the cap margin, that are covered with spore-producing cells called basidia. In boletes, a related group that includes edible porcini mushrooms and many others, the cap's underside bears pores instead of gills, openings to vertical tubes that function similarly to gills, but create different visual appearance and texture. Both gills and pores maximize surface area for spore production within the protected space beneath the cap, which shields spores from rain and dew that could cause them to clump rather than dispersing. Other fruiting body forms serve different ecological functions or reflect different evolutionary histories. Puffballs produce spores internally within a papery or leathery skin, releasing them when the skin ruptures. Mature puffballs can release clouds of hundreds of millions or even billions of spores when struck by raindrops, animal feet, or gusts of wind, with spores ejected through a small pore at the apex or through cracks that develop. The giant puffball Calvatia gigantea can grow to 50 centimeters diameter, occasionally even larger, containing an estimated 7 trillion spores, a number so large it's difficult to comprehend. If even a tiny fraction of these spores germinated and grew to produce similarly prolific fruiting bodies, the resulting fungi would cover the entire planet in a short time, illustrating why dispersal and germination success rates must be extraordinarily low to maintain population stability. Bracket fungi or shelf fungi, common on dead and dying trees, produce woody or leathery fruiting bodies that grow horizontally from tree trunks and branches, often forming overlapping shelves. These fruiting bodies contain vertical pores on their undersides, with spores produced within tubes and released downward regardless of the tree's orientation. The fruiting bodies have gravity-sensing mechanisms that ensure pores orient vertically even if the substrate is tilted or horizontal. Many bracket fungi produce perennial fruiting bodies that add new layers of pore tissue each growing season, visible as concentric rings or bands on the fruiting body surface, allowing age determination similar to counting tree rings. Some bracket fungi reach impressive sizes. The largest recorded specimen of the species, Fomitoporia ellipsoidea, found on Hainan Island, China, measured approximately 1,000 centimeters in length with an estimated weight of 400 to 500 kilograms and age of perhaps 20 years, representing an enormous investment in a single fruiting body. Morels, prized by foragers for their culinary value and distinctive appearance, produce fruiting bodies with pitted, honeycomb-textured caps atop hollow stems. The pits and ridges increase surface area for spore production, with spores formed within sac-like structures called asci lining the ridges. Morels belong to the Escamicota, one of the two major groups of fungi that produce large fruiting bodies, distinct from the Basidiomycota that includes most gilled mushrooms, boletes, and brackets. The ascomycetes produce spores differently than basidiomycetes. Spores develop inside cylindrical sacs called asci, typically with 8 spores per ascus, which eventually rupture or dissolve to release spores. In contrast, basidiomycetes produce spores externally on club-shaped structures called basidia, typically with 4 spores per basidium, which are actively discharged through a mechanism involving fluid droplet formation that catapults spores away from the basidium surface. Truffle fungi produce subterranean fruiting bodies, truffles, that don't release spores into air at all, but instead rely on animals to disperse them. Truffles develop entirely underground, often at 5 to 30 centimeters depth, and produce powerful aromatic compounds that attract animals including pigs, dogs, rodents, deer, and many others that excavate and eat the truffles, later depositing spores in their feces at new locations. The famous culinary truffles, including Perigord black truffles and Italian white truffles, can sell for thousands of dollars per kilogram due to their rarity, complex flavor and aroma, and the difficulty of cultivating them. Truffle cultivation has been partially successful with some species by inoculating tree seedlings with truffle mycelium and then planting them in suitable habitats, but production remains unpredictable and far less reliable than cultivation of conventional mushrooms, maintaining high prices. The process of fruiting body formation involves dramatic reorganization of the mycelium and represents a major developmental transition comparable to flowering in plants. In response to environmental cues, often including changes in temperature, moisture, day length, or nutrient availability, the vegetative mycelium begins aggregating hyphae into densely packed knots called primordia, visible as tiny buttons or pins on the substrate surface. These primordia then enlarge and differentiate into the various tissues of the mature fruiting body, the stem with tough, fibrous tissues providing structural support, the cap with protective skin on top and spore-producing tissues underneath, veils that protect developing structures in some species, and all the intricate anatomical features specific to each species. This development can occur remarkably quickly. Some mushrooms can grow from invisible to full size within 24 to 48 hours under favorable conditions, expanding through cell enlargement rather than cell division. The hyphae in the developing fruiting body absorb water, their cells elongate dramatically, and the entire structure inflates like a balloon being filled, allowing rapid expansion with minimal energy cost. This rapid growth explains the folk belief that mushrooms appear overnight or spring up overnight after rain. They essentially do, growing from microscopic primordia to mature fruiting bodies in a day or two when conditions trigger development. However, this rapid fruiting only occurs after the underlying mycelium has grown extensively through the substrate over weeks, months, or years, accumulating sufficient resources to support the massive burst of growth required to produce fruiting bodies. A single mushroom can produce billions of spores during its lifetime. Spore production by mushrooms is prodigious, with individual fruiting bodies of many species releasing millions to billions of spores over their lifespan, which may be only days for small, ephemeral species or weeks to months for larger, longer-lived species. This enormous reproductive output is necessary because spore survival and germination success rates are extremely low. The vast majority of spores never germinate, killed by desiccation, ultraviolet radiation, consumption by animals, landing in unsuitable locations, or failing to compete successfully with other microorganisms. Releasing billions of spores ensures that at least a few might succeed in establishing new mycelia despite overwhelming odds against any individual spore. A single large mushroom of a common species, like the meadow mushroom Agaricus campestris, might release 16 billion spores over several days of spore production, while larger species can exceed 30 billion spores from a single fruiting body. These numbers are difficult to verify precisely because directly counting billions of spores is impractical, but estimates based on spore-producing surface area, basidia density, and spore production rates yield these staggering figures. To put this in perspective, 16 billion spores represents roughly twice the number of humans currently alive on Earth. all produced by a single mushroom, perhaps 10 centimeters in diameter, requiring only days to generate this enormous reproductive output. Spore release mechanisms vary among fungal groups, but are often remarkably sophisticated, exploiting physical principles to achieve dispersal despite spores being microscopic and unable to move under their own power. In basidiomycetes with gills or pores, spores are actively discharged from basidia through a mechanism called ballistospory that involves surface tension forces. The developing spore on the basidium is connected by a thin projection called a sterigma, and at maturity, a small droplet of fluid called Buller's drop forms on the spore surface near the attachment point. Simultaneously, a different fluid droplet forms at the base of the sterigma. These two droplets suddenly coalesce, and the rapid movement of fluid creates an impulse that launches the spore away from the basidium at accelerations exceeding 10,000 times gravity, among the highest accelerations produced by any biological process. The spore travels about 0.1 millimeters, just far enough to escape the boundary layer of still air around the gill or pore and enter the free air space between structures, where it falls under gravity and is carried away by air currents. This mechanism only works in still air or gentle breezes. Strong winds would blow spores against adjacent gills or walls, preventing release. This explains why many mushrooms have complex structures that create sheltered air spaces beneath the cap where discharge can occur effectively, and why mushroom caps often have hydrophobic, water-repelling surfaces that shed water rather than allowing it to accumulate and drip, which would wash away released spores. The timing of spore release also shows sophistication. Many species release spores primarily at night when humidity is high, reducing desiccation stress, and when winds are typically calmer than during the day, allowing more effective discharge and dispersal. Once released into air, spores disperse through passive transport by wind, with dispersal distances ranging from meters to potentially thousands of kilometers depending on spore size, wind conditions, and atmospheric dynamics. Small spores, typically 5 to 20 micrometers in diameter, settle slowly through still air. Terminal velocities are only 0.5 to 2 centimeters per second for typical fungal spores, meaning they take minutes to hours to fall even modest heights of a few meters. This slow settling allows spores to remain airborne for extended periods and be carried long distances by wind. Studies using spore traps and genetic analyses have shown that some fungal species show little genetic differentiation across continental or even global scales, suggesting regular long-distance dispersal maintains gene flow between distant populations. However, most spores likely travel much shorter distances. Most spores from a given mushroom probably land within 100 meters of their source, with numbers decreasing exponentially with distance. Localized dispersal explains why fungi often show patchy distributions at landscape scales, with presence in some forest patches and absence in nearby similar-looking patches, possibly reflecting dispersal limitation rather than habitat unsuitability. Fungal conservation efforts increasingly recognize that some species may be dispersal limited, unable to colonize otherwise suitable habitats because they're too far from existing populations for spores to reach regularly, making habitat fragmentation potentially as serious for fungi as for plants and animals. Spore morphology is extraordinarily diverse, reflecting different ecological niches and dispersal strategies. Spores range in size from 2 to 3 micrometers in diameter for some yeasts to over 100 micrometers for some puffballs, with most mushroom spores falling between 5 and 20 micrometers. Shape varies from spherical to elliptical, cylindrical, spindle-shaped, star-shaped, and countless other forms. Surface texture ranges from smooth to warty, spiny, ridged, or reticulated. Color varies from hyaline, colorless and transparent, to white, yellow, pink, brown, purple, or black, with spore color often diagnostic for identifying mushroom species. Spore prints made by placing a mushroom cap gill side down on paper and allowing spores to accumulate show characteristic colors useful in identification. These morphological variations affect dispersal, survival, and germination. Ornamented spores with spines or ridges may attach more readily to animals, enhancing zoochory, animal-mediated dispersal, while smooth spores may disperse more readily by wind. Pigmented spores, particularly melanin-containing dark spores, resist ultraviolet radiation damage better than hyaline spores, potentially surviving longer in sunlight during aerial transport. Thick-walled spores resist desiccation and can survive for months or years in dry conditions, while thin-walled spores must germinate quickly after landing or they die, reflecting different life history strategies. Some spores contain oil reserves providing energy for germination and initial hyphal growth, while others have minimal reserves and must land in nutrient-rich locations to germinate successfully. Spore germination requires suitable conditions, including adequate moisture, appropriate temperature, and often the presence of specific chemical cues indicating a suitable substrate. Different species have different requirements, explaining why most spores fail to germinate even when viable. They simply never encounter suitable conditions. When conditions are met, the spore absorbs water, swells, and produces a germ tube, a hypha that emerges from the spore wall and begins growing, exploring the local environment. If conditions remain favorable and the germ tube encounters adequate nutrients, it will branch and grow, establishing a new mycelium. However, in many fungi, particularly basidiomycetes, a single germinated spore cannot produce a complete, fertile mycelium capable of forming fruiting bodies. Most basidiomycetes are heterophilic, meaning they require mating between compatible strains to produce fertile mycelia. Each spore germinates to produce a homokaryotic mycelium containing nuclei of only one mating type, and this mycelium is vegetatively vigorous but sexually sterile, unable to produce fruiting bodies. Only when two homokaryotic mycelia of compatible mating types meet and fuse do they form a heterokaryotic mycelium containing nuclei of both types, and only this heterokaryotic mycelium can produce fruiting bodies. Many basidiomycetes have complex mating systems with multiple mating type loci, creating dozens or even thousands of different mating types within a species. This means that most random encounters between mycelia result in compatible matings, increasing the probability that germinated spores will eventually produce fertile mycelia. However, it also means that isolated spores have very low probability of successfully establishing reproductive colonies unless they happen to land near other spores or existing mycelia of compatible types, further explaining the need for massive spore production. Fungi play crucial roles as decomposers, recycling nutrients from dead organic matter back into ecosystems. In terrestrial ecosystems globally, fungi are the primary decomposers of plant material, particularly woody material, and without their activity, ecosystems would quickly become clogged with dead organic matter as nutrients and carbon became locked in forms unavailable to living organisms. While bacteria also decompose organic matter, and are crucial for decomposing some materials like animal tissues. Bacteria are less effective at decomposing lignin and other recalcitrant plant compounds, making fungi irreplaceable for complete decomposition of plant litter, particularly wood. This functional importance means that despite being less visible and charismatic than plants and animals, fungi are absolutely essential to ecosystem functioning, and their loss would have catastrophic consequences for nutrient cycling, primary productivity, and the survival of other organisms. The decomposition process begins when fungal spores land on dead organic matter. A fallen log, a dead branch, shed leaves, standing dead trees and germinate, or when existing mycelia growing through soil encounter and colonize dead material. The fungus grows through the substrate, secreting enzymes that break down complex polymers into simpler compounds that can be absorbed. Different fungal species specialize on different substrates and different stages of decomposition, creating succession sequences where pioneer species colonize fresh litter or newly fallen wood, partially decomposing it and modifying its chemistry, then are replaced by secondary species that continue decomposition, eventually followed by tertiary species that complete the final stages, leaving only humus, highly transformed organic matter resistant to further decomposition. This decomposition succession is particularly well studied in wood, where distinct fungal communities succeed each other over years to decades, as a log slowly decomposes and returns to soil. Fresh wood is colonized within weeks to months by pioneer fungi that often arrive as spores while the tree is still alive, entering through wounds and remaining dormant or causing heart rot, then becoming active decomposers once the tree dies. These pioneers include many species that cause brown rot, decomposing cellulose and hemicellulose while leaving lignin largely intact, causing wood to become brown, crack into cubical pieces, and crumble when dry, though remaining relatively intact when wet. Brown rot fungi are common decomposers of conifer wood and are particularly effective at rapidly extracting energy-rich cellulose and hemicellulose, growing quickly and producing abundant fruiting bodies. White rot fungi, which decompose lignin as well as cellulose and hemicellulose, typically colonize wood later in succession, breaking down the lignin left by brown rot fungi and accessing the cellulose protected within lignin matrices. White rot causes wood to become pale or white as brown lignin is degraded, and the wood becomes soft, fibrous, and eventually spongy as decomposition progresses. White rot is the only biological process capable of completely mineralizing wood, converting all organic compounds to carbon dioxide, water, and minerals. And this process can take decades for large logs in temperate climates, or even longer in cold climates where decomposition slows dramatically. The rate of decomposition varies enormously with climate, substrate quality, and fungal community composition. In tropical rainforests, high temperatures, abundant moisture, and diverse fungal communities cause wood to decompose rapidly. Logs may completely decompose within 5 to 10 years, returning their nutrients to the soil and becoming colonized by roots of living trees that extract nutrients directly from decomposing wood. In boreal forests and Arctic tundra, cold temperatures slow fungal metabolism dramatically, and wood can persist for 50 to 100 years or more before complete decomposition. In dry deserts, lack of moisture limits fungal activity, and wood can remain relatively intact for centuries, slowly eroding through physical weathering, more than biological decomposition. Leaf litter decomposition follows similar patterns, but occurs more rapidly because leaves are thinner, less lignified, and have higher surface-to-volume ratios than wood, allowing faster colonization and enzyme penetration. In temperate deciduous forests, fallen leaves are typically colonized within days by fungi that were already present as endophytes, fungi living within healthy leaves without causing disease, that become active decomposers once leaves die and fall. These fungi rapidly break down the soft tissues, and within a few weeks to months, leaves fragment and partially decompose. Subsequent colonization by bacteria and invertebrates, including earthworms, springtails, and mites, further breaks down material, mixing it with mineral soil and incorporating it into soil organic matter. The entire process from leaf fall to complete incorporation into soil typically takes 1 to 3 years in temperate forests, faster in warm, wet tropical forests, slower in cold or dry climates. The nutrients released through decomposition, nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, and trace elements, become available for plant uptake, supporting new plant growth and maintaining ecosystem productivity. Without decomposition, these nutrients would remain locked in dead organic matter, unavailable to living organisms, and productivity would decline as available nutrient pools were depleted. Some estimates suggest that in temperate forests, decomposition releases nutrients equivalent to or exceeding the amounts cycling through living plants annually, illustrating decomposition's quantitative importance. The carbon released through decomposition as carbon dioxide returns to the atmosphere, where it's available for photosynthesis, completing the carbon cycle. Global decomposition by fungi and other decomposers releases approximately 60 billion tons of carbon annually as carbon dioxide, roughly balanced by photosynthetic uptake, maintaining atmospheric carbon dioxide at relatively stable levels over short timescales. Decomposition also creates habitat and resources for other organisms. Decomposing logs support complex communities of invertebrates, small mammals, amphibians, and plants that depend on the moist, nutrient-rich microenvironments created by fungal decomposition. Some salamander species in Pacific Northwest forests are strongly associated with large decomposing logs, using them for foraging, shelter, and breeding. Many plant species, particularly tree seedlings, establish preferentially on decomposing logs, so-called nurse logs, where they benefit from elevated positions above competing vegetation, reduced competition from herbs, consistent moisture, and nutrients released by decomposition. In old-growth forests, a substantial fraction of tree regeneration occurs on nurse logs, and the characteristic rows of large trees sometimes seen reflect establishment on now decomposed logs that arranged seedlings in lines. Decomposition also affects soil structure and properties. Fungal hyphae growing through soil and organic matter physically bind particles together, creating stable aggregates that improve soil structure, increase porosity and aeration, improve water infiltration and retention, and resist erosion. The organic compounds released during decomposition contribute to humus formation, the dark, chemically complex organic matter that improves soil fertility, water holding capacity, and cation exchange capacity. Soils rich in fungal processed organic matter tend to have better structure, higher biological activity, and greater capacity to support plant growth than soils lacking organic matter or where bacterial decomposition dominates. the importance of fungal decomposition has been recognized increasingly in forest management and ecosystem restoration. Traditional forestry practices that removed dead wood to prevent disease spread or fire hazard, and agriculture that removed crop residues to maintain tidy fields, are now recognized as potentially harmful practices that deplete soil organic matter, reduce nutrient cycling, eliminate habitat, and impair ecosystem function. Contemporary approaches increasingly emphasize retaining dead wood in forests, leaving crop residues in fields, and managing organic matter as a valuable resource rather than waste. These practices enhance fungal communities, improve decomposition services, build soil carbon, and support biodiversity, demonstrating how understanding fungal ecology leads to improved management. Mycorrhizal fungi form symbiotic partnerships with approximately 90% of plant species, facilitating nutrient and water uptake. Mycorrhizae, literally meaning fungus root, are mutualistic associations between fungi and plant roots that are among the most ecologically important symbioses on Earth, occurring in virtually all terrestrial ecosystems from Arctic tundra to tropical rainforests, from grasslands to deserts, and involving most plant families. In these relationships, fungi colonize plant roots, extending their hyphae into surrounding soil far beyond the root zone, vastly increasing the volume of soil the plant can access for water and nutrients. The fungus absorbs nutrients, particularly phosphorus and nitrogen, and water, transferring them to the plant. In return, the plant provides the fungus with carbohydrates produced through photosynthesis, typically allocating 10 to 20% or more of photosynthetic production to support mycorrhizal partners. This represents a substantial carbon cost to plants, but the benefits apparently outweigh costs in most environments, explaining mycorrhizae's near-universal occurrence. The partnership is ancient, dating back at least 400 million years to when plants first colonized land, and fossil evidence suggests that early land plants had mycorrhizal associations. Some researchers argue that mycorrhizae were essential for land colonization, allowing early plants to obtain nutrients from primitive soils that lacked well-developed mineral weathering and organic matter accumulation that characterize modern soils. Without mycorrhizal partners, early plants might have been unable to obtain sufficient nutrients to survive and reproduce on land, suggesting that terrestrial ecosystems, as we know them, owe their existence to this fungal-plant partnership. The fact that mycorrhizae are present in most plant lineages, including both ancient groups like ferns and lycophytes, and more recently evolved groups like flowering plants, indicates that the association evolved early and was retained throughout plant evolution, underscoring its importance. Two main types of mycorrhizae exist, differing in structure, fungal partners, plant hosts, and ecological roles. Ectomycorrhizae, literally outside mycorrhizae, form primarily with woody plants in temperate and boreal forests, particularly trees in families including Pinaceae, pines, Fagaceae, oaks and beeches, Bichulaceae, birches and alders, and Salicaceae, willows and poplars. In ectomycorrhizae, fungal hyphae surround root tips, forming a sheath or mantle that completely envelops the root surface, and hyphae penetrate between root cells in the outer layers, forming a structure called a Hartig net where nutrient exchange occurs. The fungi don't penetrate into root cells themselves, remaining in intercellular spaces, and the association is relatively loose. ectomycorrhizal fungi can often be cultured independently, and plants can survive without them, though with reduced growth and stress tolerance. Ectomycorrhizal fungi include many familiar mushroom-forming species, with an estimated 20,000 to 25,000 fungal species forming ectomycorrhizae. Many prized edible mushrooms are ectomycorrhizal, including porcini or boletes, chanterelles, truffles, and matsutake, which cannot be cultivated in the absence of host trees, explaining why they remain wild-harvested and often expensive. The ectomycorrhizal association is so important in temperate and boreal forests that most tree species cannot establish or grow well without appropriate fungal partners. Attempts to establish tree plantations in regions lacking compatible ectomycorrhizal fungi have often failed until fungi were intentionally introduced, either by inoculating seedlings in nurseries before planting or by adding soil containing fungi from native forests. Arbuscular mycorrhizae, literally tree-like mycorrhizae, named for the branched structures fungi form inside root cells, form with approximately 80% of plant species across most plant families, making them the more widespread type numerically. Arbuscular mycorrhizal fungi belong to a single fungal phylum, Glomeramycota, containing perhaps 300 to 500 species, far fewer than the thousands of ectomycorrhizal species, but each arbuscular mycorrhizal species can associate with many plant species, providing the broad host range explaining their prevalence. In arbuscular mycorrhizae, fungal hyphae penetrate root epidermal and cortical cells, forming highly branched structures called arbuscules inside living cells, surrounded by modified plant cell membrane, but not breaking the membrane. These arbuscules are sites of nutrient exchange, with the extensive branching creating large surface area facilitating efficient transfer. Arbuscular mycorrhizal fungi cannot be cultured independently on artificial media. They're obligate symbionts requiring living plant roots to complete their life cycle, making them difficult to study compared to ectomycorrhizal fungi that can be grown on plates in laboratories. This obligate symbiosis suggests very long coevolution, and molecular clock analyses estimate arbuscular mycorrhizal fungi diverged from other fungal lineages approximately 600 million years ago, making them ancient specialists on plant partnership. The fact that arbuscular mycorrhizae occur in earliest diverging land plant lineages, including liverworts, hornworts, and mosses, suggests this association type evolved first and ectomycorrhizae evolved later as specializations in certain plant and fungal lineages. The primary nutrient transferred by mycorrhizal fungi is phosphorus, which is relatively immobile in soil, moving primarily through slow diffusion rather than water flow. Phosphorus availability limits plant growth in many soils because although total phosphorus content may be adequate, most exists in forms unavailable to plants, and the small amount in available forms is quickly depleted in the immediate vicinity of roots as plants absorb it faster than diffusion can replenish it. This creates depletion zones around roots where phosphorus concentration drops to levels insufficient for uptake. Mycorrhizal hyphae extending centimeters or even tens of centimeters beyond depletion zones encounter undepleted soil and absorb phosphorus, then translocate it through the hyphal network to roots, overcoming the diffusion limitation. Studies using radioactive or stable isotope-labeled phosphorus demonstrate that mycorrhizal plants obtain substantially more phosphorus than non-mycorrhizal plants, often doubling or tripling phosphorus content and growth rates. Nitrogen transfer by mycorrhizae is also important, particularly in ecosystems where nitrogen limits productivity. Ectomycorrhizal fungi produce enzymes that break down organic nitrogen compounds in soil organic matter, releasing nitrogen in forms the fungus absorbs and transfers to plants. This ability to access organic nitrogen gives ectomycorrhizal plants advantages in many forests, where most nitrogen exists in organic forms unavailable to plants without mycorrhizal partners. Arbuscular mycorrhizal fungi absorb primarily inorganic nitrogen forms, ammonium and nitrate, from soil solution. But their extensive hyphal networks allow them to explore larger soil volumes and encounter nitrogen sources unavailable to roots alone. Water uptake by mycorrhizal fungi also benefits plants, particularly during drought. Fungal hyphae, being thinner than roots and capable of penetrating smaller pores, can access water in soil micropores unavailable to roots, and the extensive hyphal network can transport water from moist soil patches to roots, maintaining plant water status during stress. Field studies show that mycorrhizal plants often maintain better water status, higher photosynthesis rates, and greater survival during drought than non-mycorrhizal plants, benefits economically important in agriculture and forestry, and ecologically important for plant distribution and community composition. Beyond these direct nutritional benefits, mycorrhizal fungi provide other services, including protection against pathogens. Mycorrhizal roots are more resistant to some root diseases, possibly because the fungal sheath in ectomycorrhizae physically blocks pathogen entry, or because mycorrhizal fungi produce defensive compounds, or because altered root physiology makes roots less susceptible. Mycorrhizal fungi also improve soil structure through hyphal networks that bind soil particles, and they may produce growth-regulating compounds that affect plant physiology. The full suite of benefits explains why mycorrhizal plants typically show 10 to 50% or greater growth increases compared to non-mycorrhizal plants in natural soils, making mycorrhizae practically essential for plant success in most environments. Recent research has revealed that mycorrhizal networks can connect multiple plants, allowing nutrient and water transfer between plants through shared fungal networks. A single fungal mycelium can connect with roots of multiple plants, potentially dozens of individuals of multiple species, creating underground networks sometimes called wood wide webs. Through these networks, carbon and nutrients can flow from one plant to another, mediated by the fungus. Studies using isotope tracers have documented carbon transfer from one plant to another through mycorrhizal networks, with carbon fixed in one plant's photosynthesis, appearing in another plant within hours to days. The ecological significance of these transfers is debated. Some argue they represent meaningful cooperation or subsidy between plants, while others suggest they're byproducts of fungal physiology without adaptive significance for plants. The patterns of transfer suggest complexity beyond simple sharing. Large, well-established trees often appear as hubs in mycorrhizal networks, connected to many smaller individuals, including seedlings. Carbon transfer tends to flow from large trees to small seedlings, potentially facilitating seedling establishment in shaded understory conditions where photosynthesis is limited. However, the fungus likely controls transfer more than plants do, moving carbon to where fungal metabolic activity is highest, which may coincide with areas of active hyphal growth or fruiting body production, rather than necessarily benefiting recipient plants. Experimental manipulations severing connections show effects on seedling growth and survival, suggesting functional importance, but interpreting mechanisms and evolutionary implications remains challenging. The dependence on mycorrhizal fungi has important implications for plant ecology and conservation. Species or populations lacking appropriate fungal partners cannot establish or persist, making fungal distributions potentially limiting for plant distributions. This is particularly important for rare plants with specialized fungal requirements. Conservation may require protecting not just plant populations, but also their fungal partners, complicating conservation planning. Habitat disturbances including soil compaction, fertilization, fungicide application, and land use changes can disrupt mycorrhizal fungi, reducing plant performance even when plants aren't directly harmed. Understanding and managing mycorrhizal relationships is increasingly recognized as important for ecosystem restoration, agriculture, and forestry. Some mushrooms are bioluminescent, producing light through chemical reactions in their tissues. Among the many remarkable adaptations fungi have evolved, bioluminescence, the production of light by living organisms, is one of the most striking and mysterious. Approximately 70 species of fungi are known to produce bioluminescence, including some mushroom-forming species where fruiting bodies glow with ghostly green light visible in darkness. These glowing mushrooms occur in forests worldwide, particularly in tropical and subtropical regions with warm, moist conditions, though bioluminescent species also occur in temperate zones. The light is typically dim, requiring dark-adapted eyes to see, but can be bright enough to illuminate the surrounding area and to be photographed with long exposures. The bioluminescent reaction in fungi involves the same basic chemistry found in other bioluminescent organisms, including fireflies, marine bacteria, and jellyfish. The oxidation of a substrate molecule called luciferin by an enzyme called luciferase, producing light, oxidized luciferin, and other byproducts. The specific luciferin and luciferase in fungi differ from those in other groups, representing independent evolution of bioluminescence through convergence on similar chemical solutions. The fungal luciferin is believed to be related to caffeic acid, a compound produced by plants and found in many fungi, suggesting fungi may have co-opted existing biochemical pathways to evolve light production. The mechanism of light production involves oxidation reactions where electrons in the luciferin molecule are raised to excited states, then release energy as photons when returning to ground state. The enzyme luciferase facilitates this reaction but doesn't provide energy. The energy comes from the chemical bonds in luciferin and from the oxidation reaction itself. The light produced is typically green, with peak emission around 520 to 530 nanometers wavelength, in the middle of the visible spectrum where human eyes are most sensitive. This emission spectrum is determined by the energy gap between excited and ground states of the luciferin molecule, and similar wavelengths across bioluminescent organisms suggest convergence on wavelengths that transmit well through tissues and air and are visible to relevant receivers. The function of bioluminescence in fungi remains unclear and probably varies among species. Proposed hypotheses include attraction of insects that might disperse spores. Glowing fruiting bodies might be more visible to nocturnal insects than non-glowing ones, and insects visiting mushrooms could pick up spores on their bodies and transport them. However, observations of bioluminescent species show that many attract few insects, and some produce light in their mycelia growing through substrates where no insects would see it, casting doubt on this hypothesis as a general explanation. Another hypothesis suggests that light production is a metabolic byproduct without adaptive significance, perhaps an inevitable consequence of oxidation reactions the fungus must perform for other reasons, with light being an unavoidable side effect. Under this view, bioluminescence would be tolerated rather than favored by selection, explaining why relatively few fungal species are bioluminescent despite many producing the necessary chemical precursors. A third hypothesis suggests that bioluminescence attracts larger animals, including mammals and birds, that might disperse spores more effectively than insects, or that light might deter some herbivores or fungivores by suggesting toxicity or unpalatability. Testing these hypotheses is difficult because manipulating bioluminescence in fungi requires understanding its genetic and biochemical basis well enough to create non-glowing variants through genetic modification. Then comparing dispersal, herbivory, and fitness between glowing and non-glowing individuals, technically challenging work that has not yet been accomplished. Comparative studies across species could test predictions of different hypotheses, but the small number of bioluminescent species and their scattered distribution among fungal lineages complicates comparative approaches. Bioluminescent fungi are distributed across multiple fungal groups, indicating that bioluminescence evolved independently multiple times in fungal evolution, rather than being inherited from a single ancestral innovation. This convergence suggests that conditions favoring light production have arisen repeatedly, though what those conditions are remains speculative. Most bioluminescent fungi are wood decomposers growing on dead logs and branches in forests, suggesting that this ecological niche might provide conditions favoring bioluminescence, perhaps through producing suitable substrates for luciferin synthesis or creating environments where light production provides benefits. One well-known bioluminescent species is the honey mushroom, Armillaria mellia, and related species, which produces faint bioluminescence in its mycelium growing through wood, creating what is sometimes called foxfire, a traditional term for glowing wood. Hikers in forests occasionally encounter foxfire on rotting logs or stumps, where the glowing wood appears as eerie green luminescence in darkness. The intensity varies, sometimes barely perceptible, sometimes bright enough to read by if your eyes are fully dark adapted and you're patient. The glow waxes and wanes with fungal metabolic activity, being brightest when the fungus is actively growing and decomposing wood, dimming when growth slows. Another notable bioluminescent species is Panellus stypticus, a small bracket fungus growing on dead deciduous wood in North America, Europe, and Asia. Some populations of this species are bioluminescent, while others are not, despite being genetically very similar, suggesting that bioluminescence is controlled by relatively few genes that can be lost or inactivated without major effects on fitness. This variation within a species provides an opportunity to study bioluminescence function. If bioluminescent and non-bioluminescent populations differ consistently in dispersal, herbivory, or habitat characteristics, it would provide clues about function. However, such studies have not yet revealed clear patterns, leaving the question unresolved. In tropical regions, several mushroom-forming species produce conspicuous bioluminescence in their fruiting bodies. Mycena species in Southeast Asian forests, sometimes called electric mushrooms by locals, produce bright green light from their caps, gills, and stems, creating a spectacular display visible from meters away in dark forest understories. These mushrooms grow on dead leaves and twigs, fruiting after rains, and their luminescence may attract insects that visit the mushrooms, though whether these visits result in effective spore dispersal is unknown. Photographers seek out these mushrooms to capture long-exposure images showing dozens of glowing mushrooms scattered across the forest floor like tiny lanterns, images that have helped bring attention to these remarkable but poorly studied organisms. The genus Omphalotus includes several bioluminescent species in North America, Europe, and Asia, including the jack-o'-lantern mushroom, Omphalotus olerarius, growing on wood in eastern North America. This species produces large, orange, chanterelle-like fruiting bodies that are toxic, causing severe gastrointestinal distress if consumed, and that glow with green light, particularly from the gills. The glow is strong enough to be visible in dim light and easily photographed, making jack-o'-lantern mushrooms popular subjects for nature photographers and mycologists interested in demonstrating bioluminescence to students and the public. The species is sometimes confused with edible chanterelles by inexperienced foragers, leading to poisoning cases, though careful examination reveals differences in gill structure and habitat that allow reliable identification. Research into fungal bioluminescence is advancing with molecular techniques, allowing identification of genes involved in light production. Recent work has identified gene clusters encoding luciferase and enzymes for luciferin synthesis in several bioluminescent species, showing that these genes are clustered together in the genome and appear to have evolved through duplication and modification of existing genes involved in other metabolic pathways. This genomic organization allows coordinated expression of all genes necessary for bioluminescence, and it facilitates evolutionary changes affecting all components simultaneously. Understanding the genetic basis opens possibilities for biotechnological applications. Bioluminescence genes from fungi could potentially be transferred to other organisms to create glowing plants for ornamental purposes or for research applications, though such applications remain largely speculative. Mushroom cultivation is a global industry producing millions of tons of mushrooms annually for food. Humans have cultivated mushrooms for centuries, with the earliest documented cultivation of wood ear mushrooms and shiitake in China over 1,000 years ago, and cultivation of button mushrooms in Europe beginning in the 17th century. Today, mushroom cultivation is a major global industry, with worldwide production exceeding 12 million tons fresh weight annually and continuing to grow as demand increases for mushrooms as food, for their nutritional value, and for perceived health benefits. The button mushroom Agaricus bisporus and related species and varieties accounts for the largest share of production, followed by oyster mushrooms, shiitake, wood ear, and various specialty mushrooms including lion's mane, maitake, and enoki. Mushroom cultivation differs fundamentally from conventional agriculture of plants because fungi are heterotrophs requiring organic substrates rather than photosynthesizing and growing from inorganic nutrients like plants. Mushroom cultivation involves preparing substrates containing organic materials that fungi can decompose, inoculating these substrates with fungal mycelium, allowing the mycelium to colonize and decompose the substrate over weeks to months, then manipulating environmental conditions to trigger fruiting body formation. The entire process from inoculation to harvest typically takes 6 to 16 weeks depending on species and methods, much faster than perennial crops but slower than fast-growing vegetables, making it economically viable but requiring careful management to maintain productivity and profitability. The button mushroom Agaricus bisporus is cultivated on compost prepared from horse manure, straw, and various organic amendments including chicken manure, gypsum, and nutrients. The composting process takes several weeks, with temperature reaching 55 to 60 degrees Celsius during active composting, killing weed seeds, pest insects, and competing fungi, producing a selective medium favoring Agaricus. After composting, the material is pasteurized at 55 to 60 degrees Celsius for several hours to kill any remaining competitors, then cooled to 25 degrees Celsius and inoculated with Agaricus spawn, grain or other substrate fully colonized by mycelium of cultivated strains. The spawn is mixed throughout the compost, and under controlled temperature and humidity, the mycelium colonizes the entire substrate within 2 to 3 weeks. After colonization, the compost is covered with a casing layer of peat moss or similar material, and conditions are manipulated to trigger fruiting. Temperature is reduced to 16 to 18 degrees Celsius. Humidity is maintained near saturation, and carbon dioxide levels are controlled to stimulate mushroom formation. Mushrooms appear within 1 to 2 weeks and are harvested over a period of 6 to 8 weeks in multiple flushes or crops, with each flush producing mushrooms over a few days, followed by a week or 2 gap before the next flush. Typically, 3 to 4 flushes are harvested before productivity declines and the substrate is discarded, composted, or used as soil amendment. Total yield is typically 15 to 25 kilograms fresh mushrooms per 100 kilograms of compost, impressive productivity from a relatively small footprint. Oyster mushrooms, Pleuratus species, are cultivated on various lignocellulosic substrates including straw, sawdust, coffee grounds, and agricultural wastes, making them versatile and potentially sustainable crops that utilize materials otherwise discarded. The substrate is pasteurized to kill competitors, then inoculated and incubated at 22 to 28 degrees Celsius for 2 to 3 weeks while mycelium colonizes. Fruiting is induced by reducing temperature to 15 to 20 degrees Celsius, increasing humidity, providing light, and allowing carbon dioxide to decrease through ventilation. Oyster mushrooms fruit quickly, producing harvestable mushrooms within 5 to 7 days of initiating fruiting conditions, and multiple flushes can be harvested over 4 to 6 weeks. Yields are typically 100 to 150% of dry substrate weight as fresh mushrooms, higher than button mushrooms, making oyster cultivation economically attractive. Shiitake lentinula edodes is traditionally cultivated on logs, particularly oak and other hardwoods, inoculated with dowels or plugs colonized by shiitake mycelium, and incubated for 6 to 18 months while mycelium colonizes logs. Fruiting is triggered by soaking logs in cold water, shocking the mycelium and inducing fruiting body formation within 1 to 2 weeks. Logs can produce mushrooms for several years, harvested in flushes 2 to 4 times annually, making log cultivation sustainable and low input, but requiring patience and space. Modern shiitake cultivation increasingly uses sawdust blocks, compressed sawdust supplemented with nutrients, pasteurized, inoculated, and incubated for 2 to 3 months, then induced to fruit through environmental manipulation. Sawdust cultivation produces faster and more consistent yields than logs, though with higher inputs and shorter productive periods, representing a trade-off between traditional and modern approaches. Specialty mushrooms including lion's mane Hericium erinaceus, Maitake grifola frondosa, Enoki flammulina velutypes, and Rhizi Ganoderma lucidum are cultivated using adapted methods based on each species' biology. Lion's mane grows on supplemented sawdust, blocks similar to shiitake. Maitake grows on hardwood sawdust with specific supplements and long incubation periods of 4 to 6 months, producing large fruiting bodies exceeding 1 kilogram. Enoki is cultivated at very low temperatures, near freezing to 5 degrees Celsius, producing thin, elongated, white fruiting bodies dramatically different from wild enoki, which are short, stout, and orange-brown. The cultivated form is essentially an artificially induced developmental variant selected for appearance and texture. Rhizi is cultivated for medicinal use rather than food on various substrates, with fruiting bodies harvested immature or mature depending on intended use. Cultivation has selected cultivated strains for desirable traits including rapid growth, high yield, consistent fruiting, desirable size and appearance, disease resistance, and extended shelf life. These cultivated strains often differ substantially from wild strains, representing domestication analogous to crop domestication, though less advanced because mushroom cultivation is younger than crop cultivation. Some cultivated strains have been propagated clonally for decades or centuries, maintained in laboratories and production facilities, and these long cultivated lines may have accumulated mutations and lost genetic variation compared to wild populations. Increasingly, cultivation research focuses on maintaining genetic diversity, incorporating wild strains, and breeding new cultivars combining desirable traits, using both traditional selective breeding and modern genetic approaches. Mushroom cultivation provides economic benefits globally, offering employment and income particularly in rural areas, utilizing agricultural wastes that might otherwise create disposal problems, producing nutritious food relatively efficiently compared to animal protein, and requiring relatively small land areas compared to extensive agriculture. Small-scale mushroom cultivation is promoted in developing countries as a means of improving nutrition, providing income for women and marginalized groups, and enhancing food security. Training programs teaching mushroom cultivation have been implemented in Asia, Africa, and Latin America, with varying success depending on local conditions, markets, and support structures. Challenges in mushroom cultivation include disease management. Various molds, bacteria, and viruses can infect cultivated mushrooms or colonize substrates, reducing yields or destroying crops entirely. Trichoderma green mold is a particularly serious problem in mushroom cultivation, growing rapidly on substrates and outcompeting cultivated fungi. Managing trichoderma requires strict sanitation, pasteurization or sterilization of substrates, environmental control to favor cultivated species over competitors, and sometimes fungicide application, though organic production prohibits chemical controls. Pest insects including flies, mites, and nematodes can damage fruiting bodies or mycelium, requiring pest monitoring and management. Climate control requires energy for heating, cooling, and humidity control, creating costs and environmental impacts that cultivation operations must balance against productivity. Future directions in mushroom cultivation include developing methods for cultivating currently uncultivatable species, particularly mycorrhizal mushrooms including truffles, porcini, chanterelles, and matsutake that currently can only be wild harvested. Limited success has been achieved with some species through inoculating host plants and establishing mycorrhizal associations, but reliable, high-yield cultivation remains elusive, maintaining these mushrooms' high market values and limiting availability. Advances in understanding mycorrhizal symbioses and fungal nutritional requirements may enable breakthroughs allowing cultivation of currently wild-harvested species. Automation and robotics are being developed to reduce labor requirements in harvesting, which currently requires hand-picking each mushroom at optimal maturity, a labor-intensive task. Vertical farming and indoor cultivation are expanding, using controlled environment agriculture to maximize productivity per unit area, though with high capital and energy costs. Some mushrooms contain compounds toxic to humans, with toxicity ranging from mild gastrointestinal distress to fatal poisoning. Among the thousands of mushroom species, several hundred are known to be toxic to humans if consumed, containing various chemical compounds that cause effects ranging from mild stomach upset to organ failure and death. Mushroom poisoning is a significant public health concern globally, with thousands of cases reported annually and hundreds of fatalities, primarily from accidental consumption of toxic species mistaken for edible species, or from intentional consumption of hallucinogenic species resulting in adverse reactions. Understanding mushroom toxins, their effects, and how to avoid poisoning is crucial for anyone considering consuming wild mushrooms, as no simple rules reliably distinguish edible from poisonous species. Identification requires careful attention to multiple morphological features and ideally expert verification. The most dangerous mushrooms belong to the genus Amanita, which includes both deadly poisonous species and some edible species, creating particular risk for foragers. The death cap Amanita phalloides, native to Europe but introduced to North America, Australia, and other regions, is considered the most deadly mushroom globally, responsible for over 90% of mushroom-related fatalities. Death caps contain amatoxins, particularly alpha-amanitin, that inhibit RNA polymerase II, an enzyme essential for transcribing genes in eukaryotic cells. Without functional RNA polymerase II, cells cannot produce messenger RNA, protein synthesis stops, and cells die. The toxins particularly affect rapidly dividing cells in liver and kidneys, causing acute liver failure, kidney failure, and death within 1 to 2 weeks unless treated aggressively with supportive care, and sometimes liver transplantation. The clinical course of death cap poisoning follows a characteristic pattern that makes it particularly insidious. After consuming the mushroom, there is a latency period of 6 to 12 hours before any symptoms appear, long enough that victims may not connect illness with mushroom consumption. Then severe gastrointestinal symptoms develop, including abdominal pain, vomiting, and profuse watery diarrhea that can cause severe dehydration and electrolyte imbalances. After 24 to 48 hours, symptoms may improve, creating a false recovery period where victims feel better and may believe they've recovered. However, during this time, liver and kidney damage progresses silently, and after 3 to 5 days, symptoms of organ failure emerge, jaundice, confusion, bleeding, kidney shutdown. At this stage, treatment options are limited, and fatality rates are 50% or higher despite modern medical care. The only effective treatments are supportive care, maintaining hydration and electrolyte balance, administration of antioxidants and compounds that may protect remaining liver function, and liver transplantation if liver failure is irreversible. The dose required to cause death is remarkably small. Estimates suggest that 50 grams of fresh death cap mushrooms, approximately half of a typical mushroom, contains sufficient toxins to kill an adult, and children are more susceptible due to smaller body size. Cooking does not destroy amatoxins, which are heat-stable and remain toxic after boiling, frying, or any other cooking method. This is a common misconception that has led to poisonings, people believing that cooking makes mushrooms safe when in fact cooking has no effect on these toxins. Similarly, drying does not destroy toxins, and poisonings have occurred from consuming dried death caps months after collection. Other Amanita species contain amatoxins including destroying angels Amanita virosa, Amanita bisporigera, and related species, which are pure white mushrooms with white gills, white spores, and similar appearance to some edible white mushrooms, creating misidentification risk. These species cause identical poisoning syndromes to death caps and are equally deadly. Some Amanita species contain different toxins causing different effects. Amanita muscaria, the fly agaric with its iconic red cap with white spots, contains ibotenic acid and muscimol, compounds affecting the central nervous system, causing hallucinations, confusion, dizziness, and gastrointestinal symptoms, but rarely death in adults. However, severe poisonings can occur, particularly in children, and fatalities have been reported. Other Amanita species contain muscarine, a compound causing excessive parasympathetic nervous system activity, leading to excessive salivation, sweating, tears, small pupils, slow heart rate, and potentially respiratory failure in severe cases, though fatalities are rare with modern treatment using atropine as antidote. Another group of highly toxic mushrooms are the Cortinarius species, particularly the deadly webcap Cortinarius rubellus and similar species occurring in Europe. These mushrooms contain orellanine, a toxin that causes kidney failure, often irreversible, developing over days to weeks after consumption. The latency period before symptoms appear can be 3 to 17 days, extremely long for a poisoning, making it difficult to connect illness with mushroom consumption. Initial symptoms include nausea, vomiting, and gastrointestinal distress, followed by kidney pain, decreased urine output, and eventual kidney failure requiring dialysis or transplantation. No specific antidote exists, and treatment is supportive, but kidney damage is often permanent. These mushrooms occur in forests in Europe and Asia and have caused numerous poisonings and fatalities, though they're less well known than Amanita species. Gyromitra species, the false morels, contain gyrometrin, a compound metabolized to monomethylhydrazine, a highly toxic compound used as rocket fuel. Gyrometrin causes liver damage, kidney damage, and neurological effects including seizures. Toxicity varies among Gyromitra species, among individuals of the same species possibly due to genetic variation, and among geographic populations, creating unpredictability. Some populations of some species are consumed after parboiling and discarding the water, a process that removes much gyrometrin, but this practice is dangerous because toxin content varies unpredictably, and some individuals remain toxic even after boiling. Multiple fatalities have resulted from consuming Gyromitra, and consumption cannot be recommended as safe. Notably, some people have consumed Gyromitra multiple times without apparent harm, then experience severe poisoning, suggesting possible sensitization or cumulative effects. Many mushrooms cause gastrointestinal distress, nausea, vomiting, diarrhea, without organ damage, though these effects can still be severe and unpleasant. Chlorophyllum molybdites, the green-spored parasol, is the most common cause of mushroom poisoning in North America, causing gastrointestinal symptoms within 1 to 3 hours after consumption, but rarely requiring hospitalization and not causing long-term effects. Many other species cause similar syndromes, and individual sensitivity varies. Some people tolerate species that sicken others, possibly due to genetic differences in metabolism or gut microbiome differences affecting toxin activation or detoxification. This variability means that reports of edibility can be unreliable, and even species considered edible by some references may cause illness in some consumers. Some mushrooms contain allergens causing reactions in sensitive individuals, similar to food allergies. Shiitake dermatitis is a well-documented condition where some people develop skin rashes after consuming shiitake, caused by lentinan, a polysaccharide in shiitake that triggers immune responses in susceptible individuals. The rash typically appears 1 to 2 days after consumption, consists of red, linear streaks resembling whip marks, and resolves within 1 to 2 weeks without treatment. Cooking shiitake thoroughly reduces risk, as lentinan is partially broken down by cooking, but some people remain sensitive even to cooked shiitake. Other mushrooms can cause similar allergic reactions in sensitive individuals, illustrating that even generally edible species can cause adverse effects in some people. Alcohol-interactive mushrooms contain coprine or similar compounds that are harmless alone but interact with alcohol consumption, causing a disulfiram-like reaction. The common inky cap Coprinopsis atramentaria contains coprine, which is metabolized to a compound inhibiting aldehyde dehydrogenase, an enzyme that metabolizes acetaldehyde produced during alcohol metabolism. When alcohol is consumed within several days of eating inky caps, acetaldehyde accumulates, causing flushing, rapid heart rate, nausea, vomiting, and general malaise, similar to effects of disulfiram, a drug used to treat alcoholism. The reaction is unpleasant, but not dangerous in healthy adults, resolving within a few hours as acetaldehyde is eventually metabolized, but it can be severe enough to require medical attention. The interaction persists for several days after eating the mushrooms, so consuming alcohol even days later can trigger reactions, requiring awareness and caution. Preventing mushroom poisoning requires careful identification before consuming any wild mushroom, with expert verification highly recommended for beginners. General rules like peeling, silver spoon, or animal testing are unreliable and have led to poisonings. Toxic mushrooms don't tarnish silver. Animals can consume some mushrooms toxic to humans, and peeling doesn't remove toxins. The only reliable method is accurate species identification using multiple characteristics including cap color, shape, texture, gill attachment and color, stem features, spore color, habitat, season, and sometimes microscopic examination. Field guides and online resources provide information, but learning from experienced foragers and mycologists is invaluable. When in doubt, don't consume the mushroom. no meal is worth risking organ failure or death. If poisoning is suspected, seeking immediate medical attention is crucial, bringing the mushroom if possible for identification, or photographs if the mushroom is gone. Even if symptoms are mild, medical evaluation is important because some deadly poisonings have delayed symptom onset, and early treatment improves outcomes. Poison control centers can provide guidance, and mycological societies sometimes maintain volunteer identification services, helping identify mushrooms involved in poisonings. Clinicians treating mushroom poisonings face challenges because different toxins require different treatments, and identifying the species can be difficult if samples aren't available. Gastric decontamination through activated charcoal may reduce toxin absorption if administered early. Supportive care maintains vital functions, and specific antidotes exist for some toxins, but not others, making identification important for guiding treatment. Mushrooms have been used for medicinal purposes across cultures for centuries, with some compounds showing promise in modern medicine. Throughout human history and across virtually every culture that has encountered mushrooms, certain species have been used medicinally, reflecting observations that consuming or applying certain mushrooms produces physiological effects beyond nutrition. Traditional medicine systems in China, Japan, Korea, and other Asian countries have particularly rich traditions of medicinal mushroom use, with records dating back thousands of years describing applications for various ailments. Some of these traditional uses align with modern scientific understanding of bioactive compounds mushrooms contain, while others remain unvalidated or have been found ineffective in scientific studies, illustrating the complex relationship between traditional knowledge and modern science. One of the best-studied medicinal mushrooms is Rhiziganoderma lucidum and related species, used in traditional Chinese medicine for over 2,000 years, where it's called lingjai and considered a superior herb promoting longevity and overall health. Rhizi contains numerous bioactive compounds, including polysaccharides, triterpenoids, peptides, and other molecules with various biological activities demonstrated in laboratory studies. Polysaccharides from Rhizi stimulate immune system components, including natural killer cells, macrophages, and T cells, suggesting immunomodulatory effects that might help fight infections or cancer. Triterpenoids show anti-inflammatory and antioxidant activities in cell culture studies, suggesting potential for treating inflammatory conditions. Animal studies show that Rhizi extracts can reduce tumor growth, improve immune function, protect liver from damage, reduce blood sugar and cholesterol, and provide other benefits, supporting traditional uses and suggesting mechanisms underlying effects. However, human clinical studies of Rhizi show mixed results, with many studies being small, poorly designed, or showing modest effects. Some trials suggest Rhizi may improve immune markers in cancer patients, reduce blood sugar in diabetics, improve symptoms in people with inflammatory conditions, but effects are generally modest, and not all studies are positive. The lack of rigorous, large-scale, placebo-controlled trials makes it difficult to draw firm conclusions about Rhizi's efficacy for specific conditions. Additionally, Rhizi is generally consumed as extracts or powders rather than whole mushrooms because it's too woody to eat, and extraction methods affect which compounds are present and their concentrations, creating variability in products that complicates research and clinical use. Turkey tail Trametes versicolor is another medicinal mushroom with long traditional use and growing scientific interest. Its Japanese name kawaradake reflects its traditional medicinal use, and in modern Japan, polysaccharide extracts from turkey tail called PSK and PSP are approved as adjunct cancer treatments, used alongside conventional chemotherapy and radiation. These extracts contain polysaccharide K and polysaccharide peptide that stimulate immune function and may enhance cancer treatment effectiveness. Clinical studies in Japan show that PSK improves survival and reduces recurrence in patients with gastric, colorectal, and lung cancers when combined with standard treatments, though effects are modest and benefits are most apparent in specific cancer types and stages. These compounds are not approved in Western countries, where regulatory agencies require more extensive evidence before approval, but turkey tail supplements are widely available and consumed by cancer patients seeking complementary therapies. Lion's mane Hericium erinaceus has generated recent interest for potential cognitive and neurological benefits. Traditional use in Chinese medicine included treating digestive and cognitive disorders, and modern research suggests compounds called hericinons and erinacines in lion's mane stimulate nerve growth factor synthesis, potentially promoting neuron health and regeneration. Animal studies show that lion's mane extracts improve memory and cognitive function, reduce symptoms in models of dementia and depression, and promote nerve regeneration after injury, suggesting potential for treating neurodegenerative diseases and cognitive decline. Human studies are limited, but preliminary results suggest lion's mane may improve mild cognitive impairment, reduce anxiety and depression, and improve overall well-being, though studies are small and more research is needed. The plausibility of mechanisms and consistency of effects across preclinical studies have generated excitement, and lion's mane supplements are increasingly marketed for cognitive health. Cordyceps sinensis and related species are prized in traditional Chinese medicine as tonics enhancing energy, stamina, and athletic performance. Natural cordyceps are parasites growing from insect larvae, particularly caterpillars in high-altitude Tibetan plateau regions, and their rarity and unique life cycle make them extremely expensive, sometimes exceeding gold's value by weight. The fungus infects caterpillars, grows through their bodies, eventually killing them and producing fruiting bodies that emerge from the cadaver. The entire caterpillar fungus complex is collected and used medicinally, though most available products now use cordyceps mycelium cultivated in fermentation tanks, a more affordable and sustainable source, but possibly differing in composition from wild-collected material. Cordyceps contains nucleoside derivatives, including adenosine and cordyspin, that affect cellular energy metabolism and signaling, potentially explaining performance-enhancing effects. Studies suggest cordyceps increases ATP production, improves oxygen utilization, reduces lactic acid buildup, and enhances endurance in animal models, with some human studies showing improved exercise capacity and reduced fatigue, though results are mixed and many studies have limitations. Despite uncertain efficacy, cordyceps is widely used by athletes, and its inclusion in supplement formulations reflects popularity exceeding scientific validation, a pattern common in medicinal mushroom marketing. Beyond these well-known species, many other mushrooms are used medicinally with varying levels of traditional use and scientific support. Shiitake contains lentinan, a polysaccharide approved in Japan for cancer treatment as an immune stimulant used alongside chemotherapy. Maitake contains beta-glucans with immunomodulatory properties. Chaga inanatus obliquus, growing on birch trees in northern regions, is used traditionally and marketed for immune support and antioxidant effects, containing high concentrations of phenolic compounds and melanin. Oyster mushrooms contain statins, compounds that lower cholesterol, and consumption may contribute to cardiovascular health, though therapeutic doses would require consuming large quantities. The mechanisms underlying medicinal effects generally involve immune modulation, anti-inflammatory and antioxidant activities, effects on metabolism, and sometimes direct antimicrobial or anti-cancer activities. Many bioactive compounds are polysaccharides, particularly beta-glucans with branch structures that interact with immune cell receptors, stimulating responses that may enhance resistance to infections and cancer. Triterpenes and other secondary metabolites show diverse activities, including inhibiting inflammation pathways, scavenging free radicals, regulating gene expression, and inducing apoptosis in cancer cells in laboratory studies. However, translating laboratory effects to clinical benefits in humans is challenging. Compounds effective in cell culture may not be absorbed when consumed orally, may be metabolized before reaching target tissues, may not reach sufficient concentrations in the body, or may require specific conditions to show effects. The quality and consistency of mushroom supplements is a significant concern. Studies analyzing commercial products find substantial variation in active compound content, with some products containing little or no detectable amounts of claimed ingredients, while others contain appropriate amounts. Reasons include using inappropriate mushroom parts like mycelium grown on grain rather than fruiting bodies, poor extraction methods that don't concentrate active compounds, adulteration with fillers or undeclared ingredients, and lack of quality control. The supplement industry is less regulated than pharmaceuticals, allowing products to be marketed without proving efficacy or ensuring consistent composition, creating consumer protection issues. Some industry groups are working to establish standards and certification programs, but currently, consumers must research products and companies carefully to find high-quality mushroom supplements. Future directions in medicinal mushroom research include identifying and characterizing active compounds, understanding their mechanisms of action, conducting well-designed clinical trials, evaluating efficacy for specific conditions, developing standardized extracts with consistent composition, exploring synergistic effects of multiple compounds rather than isolated single compounds, and investigating potential applications in areas like neuroprotection, immunotherapy, metabolic disorders, and aging. Mushrooms' chemical diversity suggests they contain many compounds with therapeutic potential, and systematic exploration could yield valuable medicines. However, rigorous scientific evaluation is essential to distinguish effective treatments from ineffective or even harmful ones, protecting consumers and ensuring that traditional knowledge is validated or corrected through modern research. If sleep has found you somewhere in these quiet facts about mushrooms and their hidden networks, their spores drifting on air currents, their enzymes breaking down wood and returning nutrients to soil, then this worked as intended. If you're still awake, there's more to explore, more fungal biology waiting in the darkness of the forest floor. Rest knowing that while you sleep, fungi continue their ancient work. Mycelia are growing through soil at this very moment, extending hyphal tips through microscopic pores, secreting enzymes, absorbing nutrients. Somewhere on Earth where conditions are right, mushrooms are fruiting, pushing through leaf litter, expanding from buttons to full caps in hours, preparing to release spores into morning air. In forests, mycorrhizal networks are transferring nutrients between trees, connecting seedlings to giants, facilitating chemical communication through underground highways. Decomposers are breaking down dead wood, slowly returning fallen giants to soil, releasing carbon dioxide to air and nutrients to earth. Bioluminescent species are glowing softly in tropical forests, their ghostly green light illuminating tiny patches of darkness. Fungal spores are drifting on winds, traveling unknowable distances, landing in random locations, the vast majority perishing, but a few finding suitable microsites, germinating, beginning the cycle again. These processes operated long before humans appeared, shaping ecosystems, enabling life as we know it. They continue regardless of our attention, our understanding, our appreciation. Fungi were here first, colonizing land 400 million years before flowering plants, establishing the partnerships and processes that made terrestrial ecosystems possible. They'll likely be here last, decomposing whatever remains after other life forms have vanished, recycling molecules one final time. The next time you see a mushroom, remember you're seeing only the tip of a far larger organism, a network invisible but immense, older than the fruiting body suggests, connected to processes operating at scales from molecular to global. That mushroom is simultaneously an individual organism, a partner in symbioses, a food source for insects and animals, a habitat for microbes, a recycler of nutrients, and a disperser of genes. It's a reminder that life is interconnected in ways we're only beginning to understand, that beneath the visible world lies complexity we've barely explored. See you next time.